Okay, so good afternoon or good morning or good evening. In any case, good day, everyone, and welcome to our webinar series on molecular communication outside the box, breaking disciplinary boundaries. My name is Adam Knoll, and I'm moderating this session today. I'm the chair for the IEEE ComSoc Technical Committee on Molecular, Biological, and Multiscale Communications. And we started this webinar series as a way to bring in expertise from complementary fields to help share their knowledge and give us some good ideas for research and applications for our research field going forward. And I'm very pleased today to introduce Dr. Peter Gennemark, who's with AstraZeneca Sweden as a senior principal scientist, and he's also an adjunct professor at Linköping University in Sweden. Peter got his MSc in engineering biology from Linköping and did his PhD in Chalmers in computing science. And he's working primarily as a senior principal scientist in the area of drug metabolism and pharmacokinetics at AstraZeneca in Gothenburg, Sweden. And I'm very pleased that I've had the opportunity to work with Peter for the last few years on our own research project. And so as a research collaborator, I'm very interested in this talk today. And so I'll go ahead and hand it over to you. We will have opportunities for questions at the end. So we'll do a moderated Q&A session at the end of Peter's talk. But for now, Peter, the floor is yours. So take it away.
Thanks a lot, Adam, and thanks for having me here. It's a great pleasure. I work in a team at AstraZeneca called Modeling and Simulation. So we support the preclinical research with quantitative methods and modeling. So we basically work up till phase 1 clinical testing. So today I will talk a bit about how modeling influences drug discovery today and how it can influence tomorrow when more and more experiments are done in vitro, in advanced in vitro systems, instead of being conducted in animals. And in what ways modeling can help on this journey. So some disclosures, so I'm employed by AstraZeneca. Basically there will be 3 parts of the talk. So the traditional drug discovery workflow. So that was the disclosure. And this is the content, 3 parts. Traditional drug discovery workflow, and then microphysiological systems, and then mathematical modeling of those MPS, microphysiological systems. So when we discover drugs, we generate a lot of in vitro and in vivo data and try to integrate those to draw conclusions and pick the best compound to progress towards human. And during this process, we want to predict the human scenario. What will the dosing frequency be in human, and will this fly? Is this a future product? And then, of course, select the very best candidate to progress. So typically, we start by testing a lot of compounds, thousands of them. So with small different chemical modifications in vitro systems. For instance, to pick out the most potent compounds that can, for instance, knock down a certain target. There are also complementary cell systems to measure safety aspects. So you can remove those that have certain properties that are not beneficial. Typically then you go in vivo, for instance, in rodent, mice, or rat, and you test much fewer compounds in such an in vivo model. And in some cases, you also go to a higher species and test the efficacy, like rabbit, dog, or monkey. And then it's, of course, only a few compounds. And the very best compounds you progress to clinical trials. For most projects, of course, you will not find a good candidate and they are terminated. And from an economical perspective, you want to terminate as early as possible, because the project gets more expensive the further along to the right you go. And if we put it very conceptual, we want a drug that is efficacious and has no safety problems. And the key measure that we take is that we take blood samples and measure how much drug is in the blood for after an administration. So in this graph here, we have drug concentration on the y-axis, and we take a dose of 10. We assume you take this pill at time 0, and the concentration in blood increases, and then the drug is eliminated from the body. And you see that if the dose is 10, you nearly get over this efficacy level. So you dosed probably a little bit too less. So what's your decision? Maybe you try a greater dose. You dose 40 milligrams or whatever. And then you are in this therapeutic window where you want to be. You have some efficacy and you have no problems from safety. So this is, in this example, the level you want to have. And this is, of course, good. So this was 24 hours. And then for typical small molecule drugs, you take the next pill the next morning. And then it's the time when mathematics come into play. Because when you take the next pill after 24 hours, you still, in this example, you still have some drug left in the circulation. So you will get some kind of accumulation until you reach a steady state. And if you then simulate that, you see that the dose of 10 was actually quite good because it ends up in the therapeutic window. While if you would take the dose of 40 every day, you would have problems with safety. You will reach too high. And this is a simple illustration that mathematics and modeling is essential to draw the right conclusions and to progress the right compound with the right dosing schedule. And this is, of course, quite easy math in this case, but it can also be more complicated. So what we discussed now was pharmacokinetics. And that is basically what the body does to the drug. So it's the process of the uptake of the drugs by the body, the biotransformation they undergo, and the distribution of the drug to different parts of the body. And the drug can also be degraded to various metabolites, so parts of the drug, and how they distribute. And finally, how the drug is eliminated or excreted through the body, for instance, via the kidneys or the degradation in the liver. And what you see from a data perspective is typically those curves, that you take the drug at time 0, it's absorbed, reaches some maximum concentration, and then it's distributed to the body and it's slowly eliminated, for instance, via the kidneys. And this can quite easily be represented by a dynamical model. And typically we use differential equations with 1 or 2 state variables. And I will show some examples of that just to give you a flavor of the standard models in this field. So here's a so-called 2-compartment pharmacokinetic model for oral administration or extravascular administration. And as a cartoon, you see to the left that you have an absorption compartment, and then you have compartment 1 with volume 1 representing, you can think of it represents the circulation. And then you have a compartment 2 that you can think of represents some other compartment like the tissue or similar. And then each arrow represents some flow. And that they can be formulated by differential equations. You have one differential equation for the absorption compartment, one differential for the first compartment, and one for the second. And the initial conditions where the dose goes into the first compartment. And then if you simulate this, you will get curves like the one to the right. And since this is a 2-compartment model, compartment 1 and 2, you will basically have a biphasic elimination phase. So you have some first phase and then a second phase. You can also have 1-compartment models or 3-compartment models. And those models are extremely popular and the standard way of representing pharmacokinetics. Of course, they can be more complicated. You can have non-linearities. This standard model is linear. So if you double the dose, the concentration in plasma will double, etc. Then the other key concept is something called pharmacodynamics. That is what the drug does to the body. So that's the study of pharmacological actions on living systems. And that can, for instance, be the drug changes the blood pressure, the drug changes the food intake so that you eat less and can counter obesity. It can be a measure of pain or a measure of tumor size in cancer. So it's what happens to the body when you take the drug. And then you combine them. So for a certain drug, you can measure the PK, the drug concentration to the left. And then you can also measure the PD effect, the effect on the body. And maybe you have some standard baseline level of 1, and the drug pushes this down, and then it slowly goes back when the drug is leaving the body. And similar to PK, the PD is modeled typically by differential equations. And one of the standard ways of modeling such data is something called an indirect response model or a turnover model. And that's simply one differential equations with some input and some output, where the output is proportional to the state variable, and where the input in this case is dependent on something called C. And C is the concentration of the drug in plasma. So that's the pharmacokinetics, the PK. So we basically say that the PK, the drug in blood, drives the PD effect. It influences the PD through these differential equations, and it's saturated. It's C divided by C plus some IC50. And then you can get those types of curves. And notably, it's quite often the case that the maximum concentration in blood occurs before the maximum effects. You have a delay between PK and PD, and that is basically captured by this differential equation. And that means you have a hysteresis if you plot concentration versus PD effect. It's not a straight line, you have this loop. And if you model this, you can, of course, calculate steady state properties, etc. And this model is conceptually that you inhibit the production rate of the response R. R is then the PD marker that you measure. So a PK-PD model is then composed of a small set of PK equations and a small set of PD differential equations. So in total, you have a couple of differential equations and a quite few amount of parameters, less than 10 generally. But also here you can build more complicated models. You can go to more systems biology models with 10, 20, 30 differential equations describing more details of the biological system. But this is the most common way of representing the PK and PD. And this modeling allows us to separate system properties relating to the target biology and the turnover of the target and drug properties. So for instance, in this case, the K in and K out will be independent on the drug. They are related to the biological system. How quickly does this target turn over in the body? It can be, for instance, that you knock down a protein. How quickly is this protein produced and degraded naturally in the body? And there are drug-dependent parameters like the IC50, of course. And you can, as I said, use modeling to have concentration-response relationship at equilibrium. So when we reason about the PK and the PD, we reason about the extent and duration of response. Sorry for that. And we compare different candidate drugs. What do we want? How long exposure time do we want? Do we need to dose twice daily, for instance? We reason about the onset of action. How quickly will the PD effect come? The intensity, what is the max response we can get? Is that sufficient to treat a patient? And the duration, is the duration sufficient? Or again, do we have to dose several times a day or something like that? And then we reason about the placement and strength of this response versus concentration. How potent is our drug? Do we need a more potent drug? So this is much of the work that we do in pharma industry. So we try to contrast our compounds with competitors and different types of compounds that we have in-house in our industry. But now there's a lot of pressure, both within industry and also from regulatory agencies like FDA in US, to have new approaches to do this drug discovery and to go more and more towards in vitro systems and use less and less animals. And here are some examples with links. And then that will also, it connects to using modeling. So the idea is to use both modeling and more in vitro systems and still have good predictions of what will happen in a human. So it's a cost question and an ethical question, and it can hopefully boost the productivity. And one of those advanced in vitro systems is microphysiological systems, MPS. So it's a microfluid device that can connect multiple types of human tissues, so multiple cell types, and mimic aspects of human physiological to improve evaluations of drug responses. And the good thing here is that we can put human cells on those chips. So in contrast to animal experiments, we can rely on human cells and not on animal cells because they differ slightly. And MPS is on the more complex end of in vitro systems. Historically, one have relied on suspension. So basically a couple of cells from the same cell types in a glass well. And then one kind of 2D culture to study, for instance, acute cellular responses. And one can go to 3D spheroids. So it's sets of cells connected to each other. So it's more organ-like structures. And you can actually take different cell types and connect them into spheroids. So for instance, you can take, to represent the liver, you can take both stellate cells and hepatocytes, two of the main types of cells in the liver, and connect them into spheroids. So you get small kind of organoids. You can have them in suspension. And then you have MPS, where you put in small wells different cell types, and you can connect them by a fluid, mimicking blood circulation. And one should realize that the throughput decreases, of course, when you go here. But the physiological relevance increases and complexity increases, and that could be a better mimic of human physiology. So there are some definitions of an MPS, or organ-on-a-chip device. They should have some geometrical design and patterning. There should be some presence of flow or some microenvironmental cues like mechanical stimulations or control of nutrients, etc. And there could be sensors and online readouts, etc. There's a paper on this with specific definitions, but I will go more into examples than dive deep into those definitions. But the key point is that we want to recreate key aspects of human physiology. And that involves barrier functions. You can, for instance, have a barrier between blood and the brain, basically to protect the brain, and that can be mimicked in the MPS. You have the lung, the natural barrier, and barriers in the kidney, for instance, or in the intestine. So that can be used, for instance, to study the absorption of drugs that are taken by the oral route. And then you have the functional cell, the parenchymal cells. It could be liver cells, for instance, and here is an example of heart cells on a chip. And then, in principle, you can take different cell types and put them on the same chip. And then it opens the door to study inter-organ signaling on the very same chip. And I will give you examples of that later, where we have studied liver and pancreas cell types on the chip. And that relates to diabetes, basically, and insulin signaling. At AstraZeneca, we work with a company called Tissues in Germany, and they have their own patented MPS. And it looks like this, basically. So the size is approximately 5, 6, 7 centimeters times 2 centimeters. And there are 2 circuits on each chip. And on each circuit, you can have different cell types. And then you have a pump and the flow of medium representing circulation in human. And for instance, they have the lung liver model and lung pancreas model, etc. So it's a standard microscope glass, and you can add different cell types. It's compatible with that. And it's a pulsative flow by the pump. So it's a continuous flow of medium between the organs, allowing this crosstalk. And they can be cultivated for quite a long time. It's not only for 24 hours or 48 hours. You can actually run MPS experiments for like 3 weeks or 2 to 3 weeks. If you change the medium, so you give them fresh glucose, for instance, every 48 hours. But typically the cell cultures can survive for a long time. So that opens up for studying disease progression and more long-term PD effects, pharmacodynamic effects. And then, of course, you keep control of the sizes, how much cells you add, and how that relates to the size of the human organ. And you keep track of exactly the volumes that you have in the system and the flow rates, etc. And then, in principle, that allows you to scale to human. But it's not very simple. You actually need to think hard about this and use modeling to do that scaling. So here's some example data from this tissue chip with liver and pancreas cells. So pancreas is also called islet here. So to the left, you have single culture. So it's only one cell type on the chip. Either you have islets or liver cells. And then if you expose them to a very high glucose level, 11 millimolar, that's very high. Normal glucose is more like 5.5. Then they consume some glucose, but not very much, and they fail to go down to the normal level. If you measure insulin, you see that the islets are functional because one of the main properties of islet cell is to produce insulin when the body is exposed to high glucose. So after a meal, your body will produce a lot of insulin. And they do that, they produce a lot of insulin. But since glucose doesn't go down, they continue to produce a lot of insulin. So it's a large amount of insulin produced over those 48 hours. But then if you study the co-culture with both liver and islets on the same chip and this circulating flow so that they have some kind of contact with each other, there's a different picture. The islets will still produce insulin, but not that much as in the single cultures. Because this is enough, this insulin is enough to let the liver cells take up a lot of glucose. Because liver cells, when they sense that there are insulin in the neighborhood, they are taking up glucose so that the glucose levels decrease in the medium. And when the glucose levels decrease, there's no need to produce more insulin. So here you see an example of crosstalk. The islets on one side of the MPS produce insulin, and that insulin is going in this fluid driven by the pump to the liver cells on the other end of the MPS. And then those liver cells take up glucose, and the reduced amount of glucose in the system will tell the islets to produce less insulin. And this is published if you want to read the details. So as I indicated, it's essential to represent those systems by models because we want to translate to human. And it cannot be done by simple calculations on a small piece of paper or by just thinking. And that is because the volumes are different, the flow rates are different, and the interactions are typically involving feedback loops. And feedback loops are typically hard to translate by just thinking, because that's basically can involve non-linearities in the systems, etc. So we have this physical system. This was the ship with one compartment for islets and one for liver cells and the flow around. And we represent that by a mathematical model with two compartments, liver and pancreas, with some flow Q in between them. And we keep control of the volumes and the volume of the channel, etc. And all of this is published. This is work done together with Linköping University and a postdoc called Belen Casas. If you want to go into the detail, I will just show you the broad picture now. So we represent the system by differential equations. So that's, for instance, one differential equation for glucose in the liver compartment. And that is influenced by the dose. So every 48 hours, we give fresh glucose to the system. So by this term. And then there's a glucose flow from the islet compartment. So that's a plus. And there's a glucose flow to the islet compartment. So that's a minus. And then there are other terms that I will go into. And this is an example how data and model can look like. So this is time on the x-axis, and you note that this is several weeks, 168 hours 1 week, so this is roughly 2 weeks. You have glucose concentration on the y-axis. And this is for 3 different scenarios. So it's low glucose, normal glucose in green, and high glucose, hyperglycemia in red. And the dots are experimental data, and the lines are a model fit. And what's interesting is this zigzag pattern. And that is because every 48 hours, the system is given fresh glucose. So we feed them every 48 hours. And after feeding, the system consumes the glucose to keep the cells happy. And that's why it goes down, and then it goes up back when we feed them new glucose. You can also see that there's some progression over time, especially if you look at the red model scenario, that they tend to eat less glucose over time. So there's some kind of disease progression or something happens over time. And that is basically because the cells are exposed to so high glucose concentration that they are feeling unhappy. So you can think of this as a kind of a diabetic system. The final day here, all are given a high glucose concentration. So that's called like GTT, a glucose tolerance test, that you can also do in the clinical practice, so to test humans. So you give a high glucose and you see how quickly you can take up the glucose. So to represent the diabetic model, we use a model from the literature. That's also a differential equation model from Top et al. In 2000. So glucose uptake by the liver spheres is largely dependent on the insulin-independent glucose disposal rate. So some part of the uptake is insulin-independent, and some part is dependent on insulin. So that's represented by 2 terms in this differential equation. So now I continue to talk about this differential equation for the glucose in the liver, and I add some extra terms to capture the dependence of uptake of glucose and which part is insulin dependent and which is insulin independent, etc. And in this model, basically, glucose stimulates insulin production and insulin stimulates glucose uptake. So you have those interactions, those feedback loops in the system. And then we introduce some other state variables like insulin resistance to capture this disease progression and beta cell volume, etc., and beta cell secretion capacity. And you represent all of this by differential equations. So it's basically it's one differential equation for each of the colored boxes here. So in total, the system is described by roughly 6-7 differential equations and approximately like 10-20 parameters. And then you can make those simulations over time and test different scenarios. And for instance, we could use the model to predict response for the yellow scenario. So that then it was fitted to red and green basically, and then it predicted the yellow. That's an example of prediction using the model. So if you try to summarize this, so we show that mathematical modeling in the liver islet MPS is important to interpret the data and to translate from MPS to human. So we have created a model of the glucose-insulin crosstalk in the MPS, and that provides a mechanistic understanding of the processes underlying impaired glucose homeostasis in the system. So we measure basically glucose and insulin, and we can explain why those changes over time. And then since we represent this by parameters that are interpretable and parameters representing volumes and flow rates, etc., it is possible to make similar predictions for human by scaling to human. And here is a conceptual illustration of that we took human data from a meal response. So we had humans that had a meal, and this is average data from several patients. And you see that it's the dots of glucose. And after a meal, you have high glucose in the blood. And after a few hours, it goes back to normal. And you have corresponding insulin levels for human populations. And then the red to the right here is the translated model. So you can see that in principle, we can get the right order of magnitude for this particular system if we change flow rates, volume, and volumes. So we claim that this type of mathematical model is pivotal to interpret data from the system, which includes multiple feedback loops, and to translate flow rates, volumes, and these feedback loops from MPS to human. And of course, there is much more work to do here, but it's a nice story that goes from A to Z in this case. So in principle, if you would have such a system that is really proven to be translatable, you could, in principle, screen for new drug compounds in that system without screening in animals. And that is, of course, the long-term goal here. So the traditional way of screening in cell system, animal experiments to human to approval of a new drug could be complemented by those modeling and MPS to reduce the animal experiments. And that is what we strive for, to reduce cost, time, and have a much more ethical workflow. Now, you can also go deeper and have more advanced models to represent the systems. And that is what we have worked with Adam and Hamidreza and others at the University of Warwick. And I will show some examples of this work. And I'm not that skilled in mathematics or those equations to explain that in a good way. But I will try to explain the high level and how it connects to this picture that we have here. So the first example is work by Hamidreza Armandi, who was a postdoc in Adam Noah's lab. And he was inspired by our system and looked at those liver spheroids. So remember, liver spheroids was 3D structures of liver cells that we use in those MPS systems, represented to the left here by a figure. And and dealing with the problems of how models of this system can predict the behavior of cell cultures when it comes to signaling of certain molecules. For instance, the molecule could be glucose or insulin. And Hamidreza worked on diffusion, first of all in the medium, but also when the molecule, like glucose, entered the spheroid, and how the diffusion rate could change and what implications that would have to effective concentration. So in this case, you can think of that the path is interrupted and the molecule will stay longer time in the spheroid compared to outside. And that will lead to a net increase in the concentration on the right-hand side of the where the spheroid starts. So this is available. This work is available on bioRxiv and also under late stage review at PLOS ONE. So you can have a closer look at that. And maybe there will be opportunities later on in this series or similar to have a talk by the experts on this, on the more details. So this is on a completely different level of resolution compared to the models I've shown previously. So here we really go into the biophysics. Another similar example is done by Mitra, also in the same lab of Adam Noel. And there she considered two spheroids, where the first spheroid extracts some molecule, and the second one is the receiver. So you can think conceptually as an example that the first spheroid would be the islets, and they release insulin. And the insulin is spread in the medium, and finally they reach a spheroid of liver cells, and they are then sensing the insulin signal and can respond to that. And potential applications of this is nutrient transport or release of biomarkers, like in this case, or reception of drugs by tumors. And to the right, you have an example of molecular concentration obtained by the analysis and particle-based simulation of this receiver in this case. So in this case, the receiver will have different concentrations over time, and also depending on where, if you observe at the boundary or in the center of the spheroids. So this also takes into account the porosity of the spheroid, and she has simulated different and reasoned about different porosity assumptions. So again, this is on a biophysical and mechanistic level. And ideally, in the future, one could bridge those models to the models with ordinary differential equations that I described previously, and have even more complicated and realistic models of those systems. So some reflections and summing up. So as I tried to illustrate that there is a rapid development of alternative methods to animal experiments, and that is supported by regulatory agencies like FDA. And this can have several advantages. First of all, you use human cells. You don't study animal cells, the right cell type. You can have many sampling points. You can actually, you don't have the ethical constraints. So you can sample whenever you wish in the MPS. You can, in principle, have rich data, ethical aspects and cost aspects, of course. And to reduce cost even more, those models, of course, connected to robots, so you can automate a lot of the liquid handling, etc. And that is an ongoing process. And I claim that modeling is pivotal to scale those results to the human scenario. And another thing that I haven't mentioned is that then you can also insert missing organs, because our MPS had 2 organs. You can think of MPS with 3 or 4 organs, but you will probably not have an MPS with all human organs. But then in principle, in the mathematical model, you can insert a model representing the missing organ. And we have some pilot studies with a PhD student in Linköping that is doing this at this very moment. So we hope to be able to publish that quite soon. There's a lot of people involved in this, mainly at AstraZeneca and on the experimental side, 2 columns of names here. So thanks to all of them. We have collaboration with the company in Germany, Tissues, with Linköping University, and with Adam's group at Warwick University. So thanks to all of them. So with that, I end this talk, and I'm very happy to discuss or answer questions, etc. Thanks a lot for your attention.
Thank you very much, Peter. That was a great talk. I'm completely unbiased by saying that. It was a great overview of sort of the backgrounds and fundamentals of the pharmacokinetics and pharmacodynamics, and how the mathematical modeling has been contributing to explaining what's going on in the in these microphysiological systems. And I think you did a great summary of our work together with Mitra and Hamid Reza on modeling kind of at a more micro level the biophysics of diffusion around spheroids. So we're happy now to open up the floor to questions from the audience. You can type something in the chat, or if you'd like to raise your hand, you can also ask Peter a question directly. So we leave that up to you for any of the participants. I will remind everyone that this meeting is being recorded, so do make note of that when putting in a query. Well, perhaps I can get started, Peter, with the first question. So you were presenting some of the results of the work that we've worked on and some started publishing together, where we've really looked at kind of like a micro scale and the biophysics, and it is quite a different order of magnitude in physical scales compared to the compartmental type modeling with the differential equations with the ODEs that you were showing earlier. Do you see that as being a big bridge to gap? Like, is that are there a lot of, say, unknowns that need to be sorted out? I know we speaking from experience, I think we spent quite a while kind of working out what was an appropriate, how to define the model when looking down at a micro level. I'm curious in this more public setting to kind of get your impressions about the pros and cons of those two kinds of approaches, and what might be a good way going forward to either identify more synergies between them or kind of try to bridge that gap.
That's a really good question, and I think it partly depends on the availability of data, because those models that we use, they are informed by data, so we basically fit the models to data, while the biophysics models they're more based on first principles, so you build them from scratch, and you can basically compare them to some data, but we typically don't have data at that resolution today.
Yeah.
But the experimental techniques are evolving rapidly, so sooner or later there will be data at this level of resolution, and then it's of course easier to bridge this gap. That I think there is a gap today, at least.
Yeah.
I guess also one can think of intermediate modeling approaches. Mm-hmm. If you would consider those two that I presented now as extremes.
Yeah.
Yeah.
Yeah, no, that's certainly the case. I think there's still research proposals to be made in this domain, for sure.
Yeah.
We do have a question here on the chat from Rudolph. He's asking, how do you evaluate your systems of differential equations? Do you use some numerical environment or other means of software support?
Yeah, so we simulate them in standard softwares like MATLAB or R or something, and we fit the models to a training set of data. So generally we divide the total data we have into a training set, like 70 to 80%, and we fit the model to that. And those data are often in form of time series, which is very good when you have differential equations. And then we save some data for validation, and we then predict the validation data set and see if they agree, basically.
Right, very good. Question from Gregory. How difficult is it to prevent bacterial contamination?
Good question. I don't think we have had issues with that, but I'm not running the experiments. But I haven't heard from the experimentalists that they have had problems with that. But of course, you have to be very careful when doing the experiments.
Yes.
A lot of ethanol.
Yeah.
Yeah.
Yeah, I mean, I've been in some of the laboratory facilities in Gothenburg, and things are kept clean as far as they can be. People work in fume hoods, and a lot of standard PPE is used to help maintain the integrity of the experimental environments.
Mm.
Very good. Yeah, there's a few comments thanking from the chat. Oh, we have a hand up from Ladin. Do you want to go ahead and ask your question, Ladin?
Yeah, sure. Thank you, Adam. Thank you, Peter. Yeah, I just wonder about the system of ODEs, and I think Peter mentioned well, was it 10 to 20 parameters used in the ODE? Evaluate the equation and get the numerical values. So I just wonder where do parameters come from? And well, do you have possibility to measure or evaluate all of these parameters yourself, or you simply use some values from the literature?
So some values are from the literature or from physical characteristics of the MPS, and some are estimated from the totality of the training data. So you basically, by numerical methods, estimate those so that the fit to the data is good. So it's a combination.
Yeah, I see. Thank you.
If I could ask you a question, Peter, about the kind of the generality of the work and the models that you've been doing, because I believe most of what you showed today, you talked about glucose regulation and its applications, obviously, to metabolism. With the scope of work that you and your colleagues do at AstraZeneca or other related departments, are there similar types of systems where you apply these kinds of techniques, or is it really just the metabolic platform that you're interested in?
No, there are similar efforts. For instance, on safety, there is work on replacing bone marrow toxicity that was typically performed in animal experiments by MPS, and there's also similar modeling. And we also go more and more to kidney, for instance, and also heart. Yeah, we're trying to, but we started with insulin because it's a well characterized system. So it was an excellent starting point to study crosstalk.
Very good. Very good. Okay, I think unless we have too many more questions, I don't see anything else in the chat. So maybe this is a good point to wrap things up. And Peter, thank you again very much for your time in preparing this talk and sharing this with our community, which I know is not the field directly that you're involved in, but I think there is a lot of overlap and opportunities for synergy with the molecular communications research area. So thanks again for your time and your great presentation. And I hope that everyone in the audience enjoyed the talk as well. I'm already seeing a lot of positive feedback in the chat. And we hope this was great for everyone involved. Thank you very much.
Thank you. Thank you all. Have a good day.